So to continue with the fundamentals of the nervous system, Chapter 11b. Okay, so neurons are irritable. That means they respond to stimulus. That they respond by generating an action potential. And it's the same as pulling the trigger on a gun. You can pull a trigger gently and slowly, or you can pull the trigger quickly and violently. Either way, the bullet leaves the gun at the same way. So the impulse is the same no matter what the stimulus is. So I'm not going to worry too much about electricity, but it's basically a flow of energy. So energy is required to hold charges separate, and energy is released when charges move together. So if you can hold charges separated, there's potential energy. And letting them come together is kinetic energy. It releases energy. So that potential difference is measured in voltage. So how much is the separated charge? So the positive end of the battery and the negative end of the battery are separate from each other, and that has a voltage between them. The kinetic energy is current. Current flows to equalize the charges. Resistance is the hindrance to that flow, and we'll just leave it at that. A conductor is that which allows that to flow. So we've talked about this in the past. We talked about all of these principles in the muscular system, but there are membrane channels. There are the non-gated channels that just leak, and there are the gated channels. And the gated channels open to either chemicals, so those are the chemically gated channels, or to voltage changes, which are the voltage channels, or the mechanically gated channels. Now, the various channels are for different things, so there could be calcium channels and etc. But we're more worried right now about the sodium channels. So when they're closed, sodium can't get into the cell. When they are open, sodium can get in. Now, it can flow either way, but they're going to follow the concentration and their electrical gradients. So, because when the channels are open, the chemical and electrical gradients cause the ions to flow. And ions flowing is really current. It's electrical charges moving. So because of the sodium-potassium pump and the fact that the channels are closed, we put sodium to the outside and not on the inside. So the outside becomes relatively positive to the inside. So that's a separation of charge, which is a voltage. And I don't really care that you know that it's minus 70 millivolts, but that's what it is. It's just a potential difference. So we call it the resting membrane potential, because the inside the cell has lower concentrations of sodium than extracellular. Okay, so this is all the exact same as the muscles, and this resting membrane potential is created and maintained by the sodium-potassium pump, which uses ATP. So there's energy involved in it. So 3 sodiums get pumped out, 2 potassiums get pumped in. So potassium is found inside the cell, sodium found outside the cell. So when the permeability to the ions change of the membrane, then those ions will flow because of the concentration changes and because of the charge changes. So there's two types of signals that can happen with this depolarization, with this flow of charge. One is graded potentials, so they're short distance signals, and the other is action potential, long distance signals. When the sodium is out and not in at the resting membrane potential, we say that the membrane is polarized, and if we let the sodium in, it becomes depolarized. So the inside becomes less negative than it was, because the positive sodium ions go in. If we have a hyperpolarization, so that means the inside is more negative or the outside becomes more positive, it's harder to depolarize. It takes more to depolarize. So, and if depolarization sets off the action potential, then it's harder to reach that action potential threshold. So that's hyperpolarization. So I'm not going to worry too much about graded potential, but they're short-lived and they're local changes. They cause some depolarization or they cause hyperpolarization depending on what's going on. So the depolarized region will be, you let a little bit of sodium in right here. And then the depolarization spreads because letting the sodium in means that the voltage-gated channels next to open and more sodium comes in, and you have this spread of depolarization. So with the graded potentials, you don't reach a runaway tipping point, as it were. And all of this is just technical. You can read it if you want. It's not testable material. The action potential is when you actually get the reversal of the membrane potential, and it happens in the muscle cells and the axons of nerves. We've discussed it. It's a wave of depolarization, and this is how long-distance signals get transmitted. So at the resting state, all the gated channels are closed. So voltage-sensitive gates are closed at rest and open when they're depolarized. The inactivation gates open at rest and block the channel. So we're not worried that much about sodium channels, but sodium channels are closed at rest and they open slowly with depolarization. So because potassium is in the cell, when sodium rushes in, this inside of the cell becomes more positive, which actually drives potassium out of the cell. And there are channels that have opened to let that happen. So you get the depolarization. When we reach a threshold, it opens the next voltage-gated channels. So when we repolarize, what's happening here is the sodium channels that have opened in the depolarization phase close, but the potassium channels open. The potassium leaves, so the positive charge leaves, so now the inside of the cell is repolarized because the potassium is now the positive one outside, and there's less potassium, so it's less positive inside. So we end up restoring the inside negativity. But unfortunately, it's the wrong ions. It's now repolarized, with the wrong positives outside, and so it's with potassium outside and sodium inside. Then we have to move things back. We have to use the sodium-potassium pump to reset it. So while the potassium is outside, we have hyperpolarization. So this restores the electrical gradient, but not the ionic gradients. The ionic gradients happen because of the sodium-potassium pump, and it takes ATP to do that. So I'm not going to worry about the propagation because we talked about it already, but it moves like a wave down the cell membrane. We need a threshold. This is the voltage at which the voltage-gated channels open. How much depolarization does it take? It's like pulling the trigger. You need to reach that threshold. So if there's weak stimuli, they don't reach the threshold and you don't get an action potential. If you reach the threshold, then you get an action potential. And the action potential is the same no matter what. It's an all or nothing thing. So how do you tell the difference between a strong and a weak stimulus? If the stimulus is too weak, you don't even notice. You don't get the action potential. If it's a strong stimulus, if it is a stronger weak stimulus, then it will cause an action potential. And it'll take a long time for it to reach threshold to cause the next action potential, and it takes a long time for the third action potential. But a stronger stimulus will reach the threshold more often, and so what happens, you get a increase in frequency. So strong stimuli generates more action potentials more often than a weak one, and so we end up with frequency, and that's how we grade how strong the stimulus is. Now, it takes some time before you can have the next action potential, and this is called the absolute refractory period, because it takes time to reset the sodium and potassium balance with the sodium-potassium pumps. The more sodium-potassium pumps there are, the faster the refractory period is, the shorter the refractory period is. And I'm not going to worry about relative refractory periods. Okay, how fast does the action potential move along? Well, it moves along at fairly quickly, but it's going to vary depending on the size of the axon and how much myelination. So a large diameter axon has less resistance, and so therefore faster impulse. So large axons transmit quicker than small, fine axons. And myelination speeds things up through something called saltatory conduction. Saltatory conduction is the action potential jumps from node of Regnier to node of Regnier. Okay. It's up to 30 times faster. So because the voltage-gated channels are just at those nodes, and so the action potential jumps from node to node. It goes boom, 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 boom. It jumps from this to this. In an unmyelinated, there's more steps between here and here. So it just goes faster in the myelinated. And we're not going to worry about multiple sclerosis, as this is not a pathology course.